Hello, everyone, and welcome to today's webinar, Working with Antigen-Specific T Cells. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Lorenz Fülle, Global Product Manager for Antigen and Virus-Specific T Cells at Miltenyi Biotec. Lorenz, you may now begin your presentation.
Yeah, hi, and welcome everybody also from my side on this webinar on working with antigen-specific T cells. So in this webinar, I will quickly introduce to you our complete workflow. So from sample preparation to cell analysis to really work with the cells in a very efficient way. So actually in regard to the experimental workflow, we at Miltenyi Biotec strongly believe that the future of T cell analysis has to be more standardized, to really be able to generate more robust and also reliable results. So enjoy. So here you can see our agenda. So I will start with a short introduction and some background on antigen-specific T cells. Then I will introduce our products and solutions to work with these cells. And then I will finish with some application workflow examples. So let's start with the introduction and some background. So just a brief company presentation. So Miltenyi Biotec was actually founded in 1989 by Stefan Miltenyi himself and is until now still the largest owner-managed company in Germany with more than 3,500 employees worldwide. And what I think is really striking, 80% of our employees are still working in global research and development. And we have two main sites in Germany, headquarters in Bergisch Gladbach, and also GMP manufacturing facility in Teterow near Berlin. And we have subsidiaries in 28 countries, and we also distribute our products to additional 73 countries. So, of course, all T cells are basically antigen-specific. We talk about T cells that were activated before the analysis by a specific antigen. For example, a defined antigenic peptide, and this is really opposed to a polyclonal activation via, for example, CD3 or CD28. So by this, only T cells of a given specificity are activated, which means that the initial frequency of these cells is, of course, very rare. And of course, antigen-specific T cells and also virus-specific T cells play key roles in the immune response against viral, bacterial infections, but also in Yeah, so as I just said, I'm sorry the slides were just stuck. So they are very important, as for example, in infectious diseases, tumor immunology, but also more recently very important in the development of these new candidate vaccines against SARS-CoV-2, pharmacological screening, immunomonitoring, autoimmunity, but also allergy. So how can we analyze these? So how can we analyze these antigen-specific T cells in a bit more detail? So in general, there are two methods that can be classified into two categories. So first, there's the direct analysis of T cell specificity, for example, via MHC peptide multimers. And these multimers really provide the highest specificity to identify the rare virus-specific, tumor-specific, or other antigen-specific T cells. These peptide multimers are really working very well for CD8 positive T cells, but are limited for CD4 positive T cells. They also do not provide any information on the functional capacity of the subset, and they're also limited to a certain range of defined peptides and also MHC molecules. In contrast, there are the indirect analysis methods, which analyze more the effector functions of T cells, for example, via the secretion of cytokines or also the upregulation of certain surface markers. So this really represents an indirect identification of virus-reactive T cells after an antigen-specific stimulation. And these indirect methods, they work very well for all T cell subsets, also including regulatory T cells. They provide T cell subset-specific information, and they're also completely independent of the antigen and donor. And most importantly, these indirect analysis methods can all be analyzed using flow cytometry. So what should be analyzed when analyzing antigen-specific T cells using flow cytometry? So for example, there's CD69, which represents an early PAN T cell activation marker that is expressed in almost all activated T cells. Then we have CD25, which is also known as the interleukin-2 receptor alpha subunit, which represents a more late activation markers expressed at 24 to 48 hours after activation. In almost all activated T cells, including activated regulatory T cells. Then we have CD154, also known as CD40 ligand, which is really an ideal activation marker to analyze conventional CD4 positive T cells after about 4 to 12 hours of stimulation. Known as 41BB, which is a very suitable activation marker for CD8 positive conventional T cells. But it's also that CD137 is also expressed on regulatory T cells, but here with different kinetic after 4 to 6 hours of stimulation. And finally, of course, we can also analyze various cytokines, which can be of antigen-activated T cells, either measured in a supernatant or also on a single cell level via, for example, intercellular cytokine staining or ICS, or using our cytokine secretion assays. And of course, if you screen the literature, you might find additional markers that could be analyzed, such as, for example, CD95, Ki67, KLIG1, and others. So, as briefly mentioned before, these antigen-specific T cells have a really low cell frequency, which really represents one of the biggest challenges for a reliable analysis. So let's just have a look at the other key challenges and how we can overcome them. So, of course, with low cell frequencies, it is all the more important to really prepare the starting material in an optimal way to really prevent losing the cells in the first place. So to ensure high cell numbers, the cells can also be pre-enriched magnetically or via flow sorting, for example. Another common problem with antigen-specific T cells is often the insufficient simulation. And here, only by using powerful reagents and also really optimized protocols, the cells can be efficiently stimulated. And then furthermore, a reliable analysis is often, let's say, hampered by a high background staining, just basically because these cells are so rare. And here, reagents for specific labeling and sensitive detection methods are really important, of course. And finally, there's often a rather slow translation of these early findings into the clinic. And here, Miltenyi Biotec actually has a large portfolio for, let's say, translational research that could really help to speed up your research. So to really efficiently isolate, stimulate, stain, and analyze antigen-specific T cells, different technologies over an entire workflow to ensure successful analysis. And of course, what we think is that the right product combination here So at Miltenyi Biotec, we actually offer a complete workflow for the analysis of antigen-specific T cells. The workflow always starts with sample preparation. So here you would start from PBMC T cells from tissues. Following the isolation, the T cells would need to be stimulated either polyclonally or antigen-specific using, for example, our Peptivator peptide pools. And then following stimulation, the cells are analyzed best with the more standardized flow analysis panels using, for example, our reaffinity recombinant antibodies and our MaxQuant analyzers for flow cytometry. However, you could also think about optionally isolating T cells after preparing your sample which will increase sensitivity during analysis and also save some time. So in addition, following cell stimulation, you could also magnetically pre-enrich your antigen-specific T cells using our microbead kits. And here I would really like to enrich viable antigen-reactive T cells. And then last but not least, you could either directly after magnetic pre-enrichment also sort your cells using flow cytometer, or also after immunophenotyping sort the sequencing. And here we have the MaxQuant cytocell sorter, which really enables a sterile and also aerosol-free cell sorting of these rare antigen-specific T cells.
Okay, go ahead.
Okay, very good. So here you can just a schematic overview of our products that we offer for the analysis of antigen-specific T cells. And as you can see, we actually offer products for the whole workflow. So from sample preparation to cell culture, cell stimulation, as well as cell analysis. And if you want, you can also have a look at the respective application websites you find here on the top, and you can just click on them right now. And you can also watch our other webinars to check for other T cell subtypes. Now let's continue with our products and solutions. So for sample preparation, we could start, for example, with PBMCs. And for the isolation of PBMCs, we offer the MaxPrep PBMC isolation kit. And this MaxPrep PBMC isolation kit really provides a very fast and untouched isolation of PBMCs in about 25 minutes. And you really do not need to lyse your red blood cells or perform any density gradient centrifugation. The kit is also optimized for milliliters, and the cells have a very high purity. You will recover all of your cells, and they're also all viable. And on the graph here on the right side, you can see that the recovery of various immune subtypes found within PBMCs is at least as good, if not even a bit better, compared to conventional density gradient centrifugation using FICO, for example. Furthermore, the kit really eliminates any user-dependent inconsistencies and performs very reliable with every preparation. So it is also ideal for performing any downstream analysis, as these cells are really viable. So you could easily analyze the cells using, for example, single-cell RNA sequencing or any other downstream assays. And furthermore, the kits are also semi-automatable using the Multimax Cell 24 Separator Plus, which you can see here on the top, to really allow you to analyze PBMCs from a large cohort of samples. However, if you are interested in isolating PBMCs for larger scales in a more automatable fashion, we have also recently released the so-called whole blood PBMC kit for human PBMCs. Stopped. So again, for some cases, you might need to isolate T cells from tissues. And here we have the so-called Gentlemax tissue dissociator and also various kits. So with the Gentlemax, you can really gently produce viable single-cell suspensions and also conserve epitopes for analysis. It also works in a very reproducible fashion because it has a lot of predefined programs for more standardized tissue dissociation. And also with the Gentlemax Octo Dissociator, you are able to work very fast. So you can process up to 8 samples in parallel. And it also includes a heater module, which allows you to perfectly tune the temperature during tissue dissociation. And finally, with this sterile and closed tubes, you and your sample are completely safe during the dissociation process. And as a perfect addition, our tissue dissociation kits, which kind of combine an enzymatic treatment with the idle program from the Gentlemax, you can easily isolate T cells from tissues such as spleen, tumor, lung, but also more complex tissues like skin, brain, and also lamina propria, for example. So by the combination of this Gentlemax tissue dissociator and the tissue dissociation kits, we can guarantee you a really high yield and viability, especially for these very rare antigen-specific T cells. So for cell stimulation, we offer the so-called Peptivator peptide pools. So let's have a closer look at them. So Peptivator peptide pools are really the most efficient way to stimulate antigen-specific T cells. The Peptivator peptide pools really efficiently stimulate both, so CD4 and CD8 positive T cells, and we actually cover an extensive portfolio of virus, tumor, fungi, microbiota, and also autoantigens. The peptide pools always consist of 50 mer peptides with 11 amino acid overhangs, which actually in most cases also covers the complete sequence of the respective antigen. And they're available in research, premium, and even also max GMP grade, which then will allow a rapid translation of the early findings into the clinic. And they're also available in a high throughput version in a 96-well format. So we think that they're really the ideal stimulation reagents for both antigen and also virus-specific T cells. So as you can see here, our Peptivator peptide pools really cover the most important antigens ranging from various viruses such as CMV or an Epstein-Barr virus, but also more recently Peptivators for the SARS-CoV-2 virus. However, we also have tumor antigens such as GP100 and also melon A and the Wilms tumor protein 1, but also autoantigens like, for example, MOG and insulin. And in the plot here on the right side, you can see how efficiently the Peptivators actually stimulate both CD4 positive T cells shown on the left axis and CD8 positive T cells shown on the right axis. And here we assess the expression of gamma interferon after stimulation with the respective Peptivator in two donors. And furthermore, as I've already mentioned, we also provide the Peptivators for high throughput applications. And with them, you can really combine multiple antigens in different stripes on just one single assay plate, which will really give you some more flexibility in analyzing the cells. And in addition, I think the setup is really nice because you can stimulate, culture, stain, and analyze your cells directly on the same plate. And this will, of course, save you some time, some pipetting steps, and also the aliquoting. And in consequence, this then means that your whole experimental setup is less prone to errors. So of course, every stimulation experiment, let's say, also requires a very reliable positive control. And here our Cytostem reagent can really help you to further standardize your T-cell assays. So Cytostem has been specifically developed for rapid and efficient stimulation of human effector and mammalian T cells. Cytostem is an antibody-based reagent that actually cross-links the T-cell receptor to MHC molecules found on antigen-presenting cells. It is a non-toxic alternative to the superantigen SEB. And upon stimulation, CD4 and CD8 positive T cells will start to secrete effector cytokines such as gamma interferon, TNF-alpha, and interleukin-2, and also upregulate certain activation markers like, for example, CD137 and CD154. So again, I think this is really a perfect positive control for stimulating antigen and virus-specific T cells. So let's have a quick look on our tools that we offer for the T cell isolation and also for magnetic pre-enrichment following stimulation. So of course, flow cytometry is still the main tool for the analysis of rare T cells. However, even established methods like flow cytometry have also certain limits. Specific T cells can be extremely rare depending on the condition. And these limits were already illustrated years ago in a very nice publication by Bacher and colleagues, where the authors analyzed the sensitivity of flow cytometry and then state that flow cytometry is always limited by the number of events that can be recorded, as well as the staining background, which is typically between 0.01 and 0.1%. So in consequence, this really restricts the analysis of rare cell populations, especially with lower frequencies. So with modern flow cytometrists, you will normally be able to detect at least some cells, but there might be certain conditions where hardly any specific cell can be detected. And here you can use our MAX microbeads so more to over working with rare cells. We provide the reaffinity recombinant antibodies, which really have a very high specificity and also superior lot-to-lot consistency. And coupled with our biodyes, you can really ensure a bright staining and also a low spillover of your multi-parameter analysis. But we'll take a close look at the antibodies later on. Rare T cells. So for enrichment technology, makes the MAX technology actually so special. But actually, we just simply call it the select the best approach. So minimal labeling with no effects on cell function and a full downstream compatibility. So, but what are some of the key advantages of this column-based technology? Well, actually, the steel spheres within a column amplify the magnetic field by about 10,000-fold. So this amplification in turn then allows for really minimal labeling and also the use of this nano-sized beads, which have a size of about 50 nanometers. And in consequence, this also means that there's less epitopes are covered on the cellular surface, and also less stress is applied to the cells. Really enable a very gentle enrichment, as these steel spheres have a very cell-friendly hydrophilic coating, and there's also a large space between these steel spheres. So the cells will just pass very smoothly, actually, through the column without suffering from any negative effects. So in summary, the MAX technology is really gentle to your cells. It will not cause any unspecific cell activation. It ensures minimal labeling without building any bead aggregates. And you can also see that here on the pictures on the right side, which show microscopic pictures of T cells that were enriched with our CD3 microbeads or with microbeads from a competitor. And as you can see here on this over-labeling, which is required for column-free enrichment, really results in these bead aggregates that can already be seen in the cell culture. And also a massive over-labeling of beads on the cellular surface. In contrast, using the MAX technology, you can see that there are no bead aggregates forming in the cell culture, and there's also only minimal labeling on the cell surface. So finally, the MAX technology does not really cause any cell alterations or change the cell characteristics. And this is also why our microbeads are heavily used in clinical research and several clinical trials, for example, for cell manufacturing, and more than 55,000 patients have been already treated, approved by the FDA. So the MAX technology comes in 4 different approaches to really reflect your different starting materials. So first, we have the microbeads and the microbead kits, which are made for the positive T cell separation from, for example, PBMCs, but also various other, for example, tissues or label-free cell separation. And then second, we also have the so-called isolation kits that are made for an untouched isolation of T cells. With PBMCs or more defined in your suspension. So for both approaches, we actually cover kits for the isolation of basic T cells, but also almost any T cell subtypes from different mouse, rat, but also non-human primates. However, if you want to skip, for example, density gradient centrifugation to isolate your PBMCs, you can also use our so-called straight from microbead kits. So the straight from microbead kits are also for the positive isolation of human CD3, CD4, and CD8 positive T cells. And straight from is a very reliable way to get your T cells super fast, and it is also completely. And last but not least, we also have the so-called Max Express isolation kits, which will also isolate your T cells in an untouched fashion directly from whole blood. However, with the Max Express approach, the column-free methods. So in contrast to the other technologies, Express is not automatable, but still it is very fast and also absolutely uncomplicated to use. And actually, indeed, results after pre-enrichment using, for example, our CD154 or CD137 microbit kits are really good. So while CD154 is upregulated on activated CD4 positive T cells, CD137 is upregulated on activated CD4. Stimulated PBMCs with the CMV peptivator or left them untreated as a control. And then in both conditions, the T cells were enriched using the respective microbit kits, and then the cells were analyzed using flow cytometry. And as you can see, hardly any cell expressing CD154 or CD137 can be found in the unstimulated sample. However, following stimulation, these antigen-specific T cells that heavily upregulated CD154 and CD137 could be easily enriched. And then these isolated cells could then be easily be used in any downstream application, like, for example, TCR sequencing. And if you want, you can have a look at our nice tech paper shown here at the top, which illustrates the technology in much more detail. And furthermore, I'd like to quickly point out a technology that is absolutely unique, and these are the called cytokine secretion assays. So in contrast to intracellular staining approaches, the cytokine secretion assay really allow a viable enrichment of cytokine secreting T cells, both from mouse and human samples. And the principle is actually rather simple. So first, the secreted cytokines are immobilized on the cellular surface by the so-called catch reagent, which basically labels the cell itself with its own cytokines. And then in the second step, the immobilized cytokines are then labeled with a fluorophore conjugated antibody called the detection reagent. And by this, living cells can be detected without intracellular staining. And then optionally, in a third step, the fluorophore can be labeled, can also be used for enrichment. So with the help of anti-fluorophore magnetic beads, you can then enrich these positive cells. So consequently, our cytokine secretion assays actually come in two versions. So we have the so-called detection kits for flow analysis only, while for the enrichment and analysis of viable cytokine secreting cells, we have the so-called enrichment and detection kits. So here you can see an overview of our solutions and markers that can be used for the enrichment of antigen-activated cells. So for example, we have the CD154 microbeads that can be used for the detection and enrichment of the whole analyze the cells after 6 hours of stimulation. In contrast, we have the CD137 microbead kits where you can analyze the whole CD8 responses. For CD137 expression on CD8 positive T cells, we recommend to stimulate the cells for at least 16 hours. And finally, our cytokine secretion assay of really viable cytokine secreting cells. So now let's have a look at our tools for cell analysis. And here we have the so-called reaffinity recombinant antibodies and our biodyes. So these reaffinity antibodies have been really designed with flow cytometry in mind. They are really recombinantly engineered, highest purity and also highest lot-to-lot consistency. They all have the same universal IgG1 isotype, which really will make your experimental design much more simple. Mutated the Fc region, which will prevent the antibody to bind to Fc receptors and therefore also reduce background staining. And then coupled with our biodyes, which have a very high stain index and also low spillover, they're actually the perfect reagents for the analysis of rare cells. T cells, we actually cover all the markers that you want to analyze, and we are also always happy to help you with the design of your flow panel. So apart from the reaffinity recombinant antibodies, we also have many all-in-one solutions in the form of kits, which will help you to analyze antigen and virus-specific T cells. For example, more recently, we added the so-called SARS-CoV-2 T cell analysis kits, which allow you to stimulate and analyze SARS-CoV-2 reactive T cells in PBMC or whole blood samples. So the kits already contain a SARS-CoV-2 included a fully optimized flow panel of 8 to 9 reaffinity antibodies, depending on the version of the kit. We included a live dead dye, the viability 405, 452. We worked on a very optimized intra and extracellular staining protocol. We included all the required buffers and reagents, such as, for example, Refilin A, so you can directly start with your experiment. And the kits have a very high capacity of up to 100 tests or up to 10 to the power of 8 cells. It's also good to know that the kits really work very well with cytometer, and both kits, so the kit for PBMC as well as for whole blood samples, are also available without a peptidator peptide, and therefore could be used for the analysis of other specificities, for example. And as you can see in the plot here on the right side, specifically activated SARS, CD4 and CD8 positive T cells, analyzed here by the expression of TNF can be detected in just one sample through a combined analysis of various activation markers, like, for example, CD154, interferon, or TNF-alpha. So apart from the reaffinity antibody and the SARS-CoV-2 T cell, we also have other very convenient and also ready-to-use flow kits. For example, we have the rapid cytokine inspector, which ensures a rapid multi-parameter analysis of activated CD4 and CD8 positive T cells. And it basically does that by one-step detection of surface markers and intracellular cytokines. Then we also have the so-called MaxPlex cytokine kits for both human and mouse samples. And here, using a multiplex analysis, the kits can actually measure the concentrations of up to 12 or 10 different cytokines in cell culture supernatants. And the technology is based on our MaxPlex capture beads, which will basically work on any standard flow cytometer. And then finally, we also have the so-called 8-color immunophenotyping kit, which enables an optimized flow-based evaluation of various leukocyte subsets, such as, for example, monocytes, neutrophils, eosinophils, T and B cells, as well as NK cells. And our flow portfolio is rounded off with our flow analysis and sorting instruments of the so-called MaxQuant series. So first, we have the MaxQuant analyzer, which is a really powerful benchtop flow cytometer with up to 16 optical parameters. And really the clue here is that it allows for fully automated acquisition using a robotic sample collector and the so-called express modes. So these express modes you can imagine as some kind of software modules that are pre-installed on your MaxQuant and allow an automated analysis of your flow analysis results. And furthermore, the instrument is really small and you can easily place it basically under every flow hood. And second, we also have the MaxQuant Tidal cell sorter, which opposed to classical droplet sorters, allows for a more gentle cell sorting approach and higher viability, even for the rare and sensitive cells. And therefore, it uses a microchip sorting technology to sort your cells in a mechanical way. So furthermore, this closed and sterile cartridge system really makes the Tidal the ideal instrument for basically any GMP environment. And here I would like to have a short look, especially on the MaxQuant Tidal cell sorter. So with the really unique capabilities like this gentle cell sorting and the closed and sterile cartridge system, the Tidal is really good for the isolation of rare T cells. So for example, for the isolation of CAR T cells, regulatory T cells, but also CAR Tregs, antigen-specific T cells, but also, of course, virus-specific T cells. So let me just briefly point out that the Tidal is already being part of several clinical trials, actually involving the analysis of tumor-specific cells, as can be seen here on the list on the right side. Okay, let's now come to some application workflow examples in the last part for today. So I would like to start with the rapid cytokine inspector, which will help you to simultaneously stain T-cell activation markers such as CD154, as well as various effector cytokines, actually idle to detect cells with a relatively low frequency of, let's say, 0.1 to 0.01% of the total T cell. So here on the right side, you can see a typical application workflow. So first, you would need to isolate your PBMCs, then you stimulated the cells either specifically with, for example, our peptide pools, or polyclonally with, for example, anti-CD3, CD28. Then you would stain your surface markers for about 10 minutes, fix the cells for 20 minutes, and then perform a permeabilization and intracellular staining for about 20 minutes. So this whole protocol of the rapid cytokine inspector takes only 40 minutes before you can go ahead and start analyzing your cells using flow cytometry. And of course, the rapid cytokine inspector is also compatible with our Max Express modes for the Max Quant analyzer. So the rapid cytokine inspector could be, for example, used for fast and automated assessment of antigen-reactive CD4 and CD8 positive T cells from PBMC samples. It could also be used for the immunomonitoring of antigen-specific T cells, but also, for example, to test the eligibility of a leukapheresis product by, for example, assessing the presence of, for example, virus-specific T cells. So actually, in this example here, you can see how the rapid cytokine inspector compares to standard protocols for intracellular cytokine staining. So here, PBMCs were stimulated with the CMV peptivators PP65 and IE1 or left untreated as a control. And then the frequencies of CMV-specific CD4 and CD8 positive T cells were analyzed. And as you can see, the frequencies are completely comparable between two approaches, with the striking difference that the rapid cytokine inspector protocol only takes about 45 minutes to complete. So in the second example, human PBMCs were again incubated with the CMV PP65 peptivator for 6 hours. And then after 2 hours, we added Brefildin A, and then we analyzed the cell. However, here we use the rapid cytokine inspector to analyze the expression of gamma interferon and interleukin-2 on these antigen-activated CD8 positive T cells. And as you can see here, we successfully detected both interferon gamma positive single positive cells, as well as interferon gamma and interleukin-2 double positive cells. So next, I would like to quickly introduce the so-called rapid ARTO protocol, which stands for Rapid Antigen-Reactive T-Cell Enrichment. And rapid ARTO is actually what I think the perfect solution for the analysis of rare antigen-specific T cells, as it combines cell stimulation, surface marker staining, intracellular cytokine staining, and an enrichment step based on T cell activation markers, such as, for example, CD154 or CD137. However, what I think is really cool about rapid ARTO is that permeabilization and intracellular cytokine staining can be performed directly on the column. And it's actually perfect for very low target cell frequencies of 0.01 to actually 0.001% of the total T cell population. So, and rapid ARTO can be, for example, used for the identification of rare virus-reactive T cells, such as, for example, SARS-CoV-2 reactive T cells, but also for the detection of rare tumor-specific T cells, for example, Wilms tumor protein 1 specific T cells. It can also be used for detailed analysis of the complete rare CD4 positive and CD8 positive T cell repertoire, and also be used for cytokine profiling, for example, of fixed fungi or tumor-specific T cells. So let's have a quick look at a virus-specific example where the rapid ARTO protocol was applied. So here, PBMCs from a COVID-19 survivor and a healthy donor were stimulated with a mix of SARS-CoV-2 peptivators, a mix of influenza peptides as a positive control, or also left untreated. And as you can see in the middle panel, the rare SARS-CoV-2 reactive T cells were successfully enriched using CD154 and could then be easily visualized in flow cytometry analysis. And the data was actually provided by the courtesy of Alexander Scheffold and also Petra Bacher from the University Clinic Schleswig-Holstein in Kiel, Germany. And if you want to find out more about the Rapid ARTO protocol, please have a look at their publication here. And also in the second publication here on the lower right, the Rapid ARTO protocol was used for the enrichment of SARS-CoV-2 reactive CD8 positive T cells. So in this last example for today, I would like to quickly show you how you could use our MAX technology to enrich antigen-specific T cells via peptide MHC multimers. So again, we can combine cell separation and flow staining directly in one step. And as you can see in the workflow here on the right side, we would first need to stain the cells using a highly specific MHC peptide multimer that is coupled to fluorophore, such as, for example, APC or PE. And then in the second step, we label the cells with anti-fluorophore microbeads. And this way, we'll then specifically enrich only those cells that express the distinct T cell receptor. And then following elution, we can directly analyze the enriched cells using flow cytometry. So this method is really ideal for an efficient in-depth ex vivo analysis of these rare antigen-specific T cells. Furthermore, you could also use this approach for the enrichment and multi-parameter analysis of ultra-rare antigen-specific CD4 and CD8 positive T cells, for example, to analyze naive tumor-specific T cells. It will ensure flexible enrichment via these anti-flow microbeads. So this way, the approach is also adaptable to MHC2 multimers and CD4 positive T cells. And we can combine MHC multimer staining also with intracellular cytokine staining. And here we have developed an optimized protocol that you can click on here. So in this last example, we used in a publication from Schmid and colleagues published in Immunology in 2015, naive Wilms tumor protein 1 specific CD8 positive T cells were analyzed in healthy donors. So this Wilms tumor protein 1 is a transcription factor that is expressed at high levels in several hematological, but also some solid tumors. It is highly oncogenic and actually ranked as one of the top tumor-associated antigens for cancer immunotherapy. However, the authors here found that relatively little is known about the presence and also functional relevance of these WT1 specific T cells in healthy donors. So in this study, naive PBMCs were first stained with WT1 tetramers coupled to APC and PE, and then anti-PE microbeads were used to specifically enrich WT1 specific CD8 positive T cells before the analysis using flow cytometry. So for the analysis, they then actually employed two internal control mechanisms to control for non-specific background staining. So as already said, they performed this dual labeling with tetramers conjugated to PE and APC, and then analyzed only the double positive events. And second, they also analyzed the tetramer staining on CD4 positive T cells as an internal reference standard. But as you can see, these ultra rare WT1 specific CD8 positive T cells could be successfully enriched using the anti-PE microbeads. And then further analysis actually revealed that the cells displayed a naive CD45 RA positive, CCR7 positive phenotype. So the authors actually conclude that the majority of WT1 specific CD8 positive T cells detected in healthy donors originate from a naive T cell pool. And if you want to have more details, you can also have a look at their publication here in the lower right corner. So this was our last example for today. And again, I'm really sorry that I lost the connection in between, but let me just briefly sum up the main things of today. So really the key for the analysis of rare T cells is to prevent cell loss from early on, to pre-enrich your cells, to use the most efficient tools for stimulation, and also make sure that they have a very low background staining. And we at Miltenyi Biotec actually offer a complete workflow for the analysis of antigen-specific T cells, which includes an optimal PBMC isolation with our PBMC isolation kits, tools for the efficient sample preparation with the Gentlemax tissue dissociator, and also our tissue dissociation kits, potent T cell stimulation with the Peptivator peptide pools, efficient pre-enrichment of antigen-specific T cells using CD137, CD154 microbead kits, cytokine secretion assays, as well as anti-fluorophore microbead kits, You need a low background staining, and you can achieve that with our reaffinity recombinant antibodies. We offer many all-in-one flow analysis kits, both for surface marker staining as well as cytokine analysis, such as, for example, the rapid cytokine inspector, as well as the MaxPlex kits. And using the MaxQuant analyzer and the Titer cell sorter, you can ensure an automated flow analysis and a very gentle cell sorting. So just please recognize the displayed disclaimer. And with this, I come to an end, and I would really like to thank you very much for your attention and for staying on. And I'm happy to take any of your questions, either now or also later.
Thank you, Lorenz, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. All right, let's go ahead and get started. Okay, so your first question, let's see, is do you also have peptivator peptide pools for the new SARS-CoV-2 variants of concern?
Oh yeah, that's a very good question. So actually, in fact, we just released several peptivator peptide pools for all the new mutations that are that were originally discovered, for example, in the UK, South Africa, Brazil, or also most recently in India. And for these mutation pools, we actually always offer two pools. One is the mutation pool, which covers the mutated sequence, and one is the respective wild-type control pool. So really, by performing separate simulation with the mutation and the reference pool, you can then detect and analyze this mutation-specific T-cell responses.
All right, great, thank you. Okay, your next question. Can the SARS-CoV-2 T-cell analysis kits also be used for the analysis of other antigen-specific T-cells?
Oh yeah, that's also an interesting question. So as I've said in my presentation, we've also just released a version of the kit that does not include a peptivator peptide pool. So in this kit version, you could just use your own stimulation control to stimulate, for example, tumor-specific T cells, and then just use the included reagents and the included flow panel to analyze antigen-specific T cells.
Okay, great. Thank you. And your next question, could you please quickly explain how the MaxPrep PBMC isolation kit works?
So the isolation of PBMCs using the kit is actually performed with only one labeling step, and also in a two-step separation procedure. So during the first isolation step, erythrocytes are aggregated and just sedimented, and then in the second step, the PBMCs are isolated by the depletion of non-PBMCs, so neutrophils, eosinophils, platelets, and residual erythrocytes. So by this, you will isolate untouched PBMCs.
Okay. Is there a surface marker that could be used for the enrichment of regulatory T cells?
Oh, that's an interesting question. So for the enrichment of regulatory T cells, we actually recommend to also use CD137. However, here, CD137 is expressed on activated CD8 positive T cells as well. So if you want to specifically enrich regulatory T cells, you should only stimulate your cells for about 4 to 6 hours. Then CD137 is expressed on regulatory T cells, while if you stimulate your cells for an extended period of time, you will also enrich CD8 positive T cells.
Thank you, Lorenz. Looks like we have time for one last question here. How many samples can be analyzed using the SARS-CoV-2 T-cell analysis kits?
So the kits are actually optimized to perform 100 tests. However, for the analysis of one donor, we strongly recommend to always include a separate negative and positive control. So in total, this means you could analyze up to 33 donors and always have a positive and a negative control included. Well, thank you again, Lorenz, for your time today. And we would also like to thank LabRoots
and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. And before we go, I'd like to thank the audience for joining us today and for their interesting questions. I know we ran out of time today, so any questions that were submitted that we didn't answer today or those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. And we encourage you to share that email with your colleagues who may have missed today's live event. All right, well, until next time, thank you and goodbye.